Microbial fuel cell, Nature's Energy, a clear answer to fuel deficiency by Molly Eron. My experiment was to address the problem of our continued depletion of non-renewable resources. I do this in my experiment by advancing microbial fuel cells, which harness the power of respiring microbes in the soil. A microbial fuel cell is a device that converts chemical energy from microbes in the soil into electricity through the oxidation and reduction reactions that occur naturally. In the soil, the benthic organisms need nitrogen, phosphorus, micronutrients, and carbon dioxide to feed off of, and compost provides this. MFCs located on wastewater treatment centers would be highly efficient. Adding compost or natural waste can efficiently boost the energy output in an MFC while using materials that are both easily accessible and sustainable. I hypothesize that if the compost of an animal or vegetable waste is added to the soil in the anode chamber of the microbial fuel cell, then the voltage output of the microbial fuel cell will increase from the control trial. In a microbial fuel cell, the wastewater and soil is found in the anode chamber. The bacteria release their electrons into an electrode in the anode, and the electrons travel across a wire under a load into the cathode. There, the electrons combine with protons and oxygen to form water. As electrons travel from the anode to the cathode, they create a current, which is where the voltage is measured. The materials needed to construct 5 microbial fuel cells include 10 containers, 5 PVC pipes, 10 plastic flanges to match the pipe width. It would also include carbon cloth and epoxy or sealant. You need resistors, if you feel necessary, 2 wires per fuel cell, and a digital voltmeter for the recordings. To make the salt agar, you need agar, sea salt, and water. The anaerobic mud samples come from a lake or creek. You would also need salt water to put in the cathode. And finally, you need a sample of each composting material. I chose mushroom soil, poultry manure, and also the addition of compost accelerator. Here are the detailed procedures of my experiment. I gathered the materials, collected a benthic sample from a creek or lake, connected 2 containers with a PVC pipe, formed the salt bridge, constructed the electrodes, assembled the MFC, and began running the microbial fuel cell. This is a step-by-step representation of what I did in my procedures through images. For the experimental design, the independent variable is the type of compost that is added with the mud into the anode chamber of the MFC. The dependent variable is the voltage output the MFC produces in millivolts. The control group is a baseline MFC that contains only mud in the anode chamber. Controlled variables include the amount of compost added, the size of the MFC, the electrode shape and size, the light and heat exposure, time of production, and data collecting. I included 2 trials with the 5 variables of mud, mushroom soil, poultry manure, mushroom soil with compost accelerator, and poultry manure with compost accelerator. The control environment of the microbial fuel cell, where only mud was added, the 2 different trials showed varying trends. The next variable placed inside the MFC was the composted mushroom soil. Another compost that was tested in the MFC was the poultry manure compost. The results changed with the addition of both mushroom soil and compost accelerator. Both trials were similar in this test. For the final compost addition, poultry manure and compost accelerator were added to the MFC. Graph 1 of Trial 1 shows that mushroom soil with accelerator performed the best throughout the week, but composted mushroom soil proved to be the highest on the final day. Graph 2 of Trial 2 shows that poultry manure with accelerator performed high above the rest. Graph 3 of the overall average of the trials shows that all composts performed better throughout the week than the mud. My hypothesis was proved correct as the addition of compost into the anode chamber increased the voltage outputs of the microbial fuel cells, but the ideal compost would be poultry manure with accelerator. Then would come the mushroom soil with accelerator, and finally composted mushroom soil in third. The cause of the increased voltages. The accelerator in the MFCs produces higher results than the regular composts because the accelerator speeds up the decomposition of organic matter in the container, and the microbes can then consume and grow at a faster rate. When compost is added to the soil, the organic component substances within help to sustain healthy bacteria, provide growth enhancers, discourage the growth of harmful organisms, and encourage the growth of helpful organisms. Digging up mud from a marshy area makes it difficult to determine the contents of the mud and can lead to the results of Trial 1 versus Trial 2. Compost material and compost with accelerant provide more consistent and predictably better results.